Good day and welcome, everyone. I'm Jack Van Horn from the University of Virginia. Please allow me to welcome you to today's edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. It is my honor and privilege today to welcome our speaker, Dr. Shefali Jesti, Associate Professor in Residence in Psychiatry, Neurology, and Pediatrics at the University of California, Los Angeles, David Geffen School of Medicine. Dr. Jesti is a behavioral child neurologist specializing in autism and related neurodevelopmental disorders. She earned her bachelor's degree in philosophy from Yale University in 1997 and her MD from Harvard Medical School in 2002. Dr. Jesti then completed a residency in child neurology and a fellowship in behavioral child neurology at the Children's Hospital of Boston. She's conducted postdoctoral training in developmental cognitive neuroscience with the great Charles Nelson at Harvard Medical School, where she gained expertise in the use of high-density electroencephalography to characterize functional brain development in infancy and early childhood, particularly as it informs atypical development. Dr. Jesti's research is focused on developing methods to improve precision in the diagnosis and treatment of neurodevelopmental disorders from early infancy through late childhood. Among her many accomplishments, she holds several national and international leadership positions, including the Board of Directors of the American Brain Foundation, the Board of Directors of the National Organization for Rare Disorders, the Board of Directors for the International Society for Autism Research. In 2019, she became the chair of the International Baby Siblings Research Consortium. And also in 2019, she was awarded the Presidential Early Career Award for Scientists and Engineers for her innovations in research in early predictors and intervention for genetic neurodevelopmental disorders. Today's presentation is entitled Clinical Trial Readiness for Neurodevelopmental Disorders on the Road to Precision Health. As always, we are streaming this lecture live for recording via YouTube. If you are watching on YouTube, thank you so much for joining us today. Also, our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are encouraged to submit any questions for Dr. Jesti via the chat feature of their Zoom sessions. I'll synthesize all these questions and ask them on your behalf during the last 10 minutes or so of the lecture. And with that, welcome, Shefali. We are so excited to hear about your lecture. You're muted, Shefali.
You would think after a year of Zoom, I wouldn't do that. So thank you, Jack, and everyone for inviting me, and for all those of you who are joining on I was going to say Friday morning, but you're on the East Coast, so a little later for you, lunchtime break. But yeah, thanks so much for inviting me, and I'm really excited to speak with you today. I love sharing our work with folks with different backgrounds. And I think you, as many of you are data scientists, are really critical for the kind of work that we do. I'm going to keep this talk kind of high level and really give you a big picture with regards to sort of where we are in neurodevelopmental disabilities and our efforts to really improve precision health and clinical trial readiness. But I hope that as I do that, many of you who are really, again, using your expertise to think about how we creatively can use our big data systems and our really complex data sets to help answer some of the questions that we need answered. Hopefully that'll help, motivate you, inspire you to kind of get involved with this field if you are not already. And then I'm going to make sure I keep some time for questions at the end, because I'm not going into the weeds with our methods, especially with the EEG methods, but happy to answer questions about that at the end as we go. So anyway, thanks again for inviting me. And yeah, so I'm a pediatric neurologist. These are my disclosures, none of which really have any bearing on what I'll talk about today. But I'm a peds neurologist. As Jack said, I trained at Boston Children's Hospital and then spent some time working with Chuck Nelson to really learn about, what are the challenges and approaches that we can take to understanding and studying early brain development? Because we know that brain development is what has gone in some ways sort of askew in neurodevelopmental disabilities. And so I learned some methods, mostly EEG, and then moved to UCLA in 2010 to be a part of this large center that we call CART, Center for Autism Research and Treatment. And what's pretty cool about CART is that it's a very integrated, multidisciplinary team. where we have experts, not just in sort of research and early markers and genetics and biomarkers, but also experts in clinical care and treatment. And a lot of the work that we do is to integrate those disciplines to really ask these important questions about how do we improve diagnosis and treatment for neurodevelopmental disabilities. And you'll hear a tiny bit about this clinic that I started that is a multidisciplinary program called CARING, which stands for Care and Research in Neurogenetics. And it's a clinical platform where we evaluate and then help to manage kids and adults who have genetic causes for their neurodevelopmental disability. So what I'm going to do today is just go through a few things. So I'm going to start by giving you this big overarching view on how we think about precision health in neurodevelopmental disorders. It's a big buzzword in the field of medicine right now across the board, and it's become a reality in neurodevelopmental disabilities, which is super exciting. I'm then going to sort of shift a little bit and talk about, sort of a reality check on this excitement about precision health, and talk about, what do we need to do from the standpoint of clinical trial readiness to make sure that when the therapeutics are developed, we're ready with the trials. And that's really the bulk of my research is in that space. And then finally, just given the time that we are in during COVID, I am going to talk a bit about, just quickly, 5 minutes, about considerations that we need to be making both in clinical care and research in neurodevelopmental disabilities during this challenging time in COVID, which seems to never be ending, especially in Los Angeles, where our rates keep rising. Okay, so just to get us all on the same page, since I know many of you are not clinicians, what are neurodevelopmental disorders? So these are diagnoses, importantly, that are based on behaviors. So they're based on a list of symptoms that are in the chapter of neurodevelopmental disorders in our Diagnostics and Statistics Manual. The newest version is DSM-5. What's important about these behaviors is that they emerge early in development. And there's a lot of overlap in symptomatology. So the main diagnoses you'll hear about with NDDs are things like global developmental delay, autism spectrum disorder, and intellectual disability. There's others like ADHD and social communication disorder, but it's really global delay, intellectual disability, and autism that you'll hear me talking about. And you'll hear me talking about work that's really focused on autism, but I just want to emphasize that a lot of these developmental disabilities overlap. What's important to know, though, is that there's a tremendous amount of heterogeneity in these different diagnoses, right? So, there's a famous quote that if you've met one child with autism, you've met one child with autism. But the reality is that there is a lot of variability in how kids get to these list of symptoms, if you will. And part of the reason for that heterogeneity is that there's a lot of etiological heterogeneity. Now, there's a high genetic liability, and we'll talk about that, but there's many different biological and even environmental ways that one can arrive at these developmental disabilities. And so that really is actually a challenge for us when we're thinking about designing treatments, or even, trying to identify clinical patterns that we might want to target, again, with treatments or with evaluations. So despite all that heterogeneity, though, our treatment model has really historically been this. So I see a kiddo in clinic, say with an autism diagnosis, right? So autism, you have core symptoms or deficits in social behavior, as well as the presence of repetitive behaviors and restricted interest, or RRBs. So I diagnose a child with autism, and then I essentially recommend a treatment. And that treatment might be a behavioral intervention, or it might be a medication. And then basically what I'm looking for is whether those basic core symptoms have changed. So I'm measuring quite crudely often that outcome, which would be, again, those core symptoms. And again, the treatment could be a medication or a behavioral intervention, but it feels crude. And as a clinician who's trying to be precise in diagnosis, it becomes a bit frustrating at times to then say, I've made this really precise diagnosis, and now I'm just going to have you go and, access whatever services your regional center or your school system can provide you. And we'll hope that we see a change in your outcome. We'd like to move towards what we consider precision health. And I'm going to sort of operationalize precision health in a few different ways for you guys. But, the idea of precision health is that as we start understanding causes a bit better, and we identify specific clinical symptoms that may be more proximal to the cause, we then can identify treatments that actually might be targeting those specific symptoms or even the cause. And then our outcomes that we're measuring might be a little more specific, again, to that subgroup or to that cause, and ideally would be evidence-based. So we actually have data on what those outcomes might be that might be measurable. So this is kind of the basic rubric, really simplistic, of where we'd like to move from the standpoint of more precision health in neurodevelopmental disorders. I love this quote from Obama, timely this week to sort of quote him, from his State of the Union address, many years ago, where he really put forth a plan for a large federally funded initiative towards precision health that was largely sponsored by NIH, where he defined precision health as delivering the right treatment to the right patient every time. And I think that's definitely a goal in our case as well. I'll add a few caveats. So at this point, the one caveat I'll add to that in neurodevelopmental disabilities is that we also want to make sure that we provide the right treatment to the right kid at the right time. And timing is often, we think, key in these developmental disabilities where we think that changes are occurring even in utero, right? So we'd like to try to target treatments as early as possible, which is challenging for a variety of reasons, as we'll talk about. So how do we even start thinking about precision health and neurodevelopmental disorders? Like, where do we look given this big heterogeneous behavioral set of conditions that we have? And this is where genetics has actually been incredibly transformative. And again, I'm going to summarize a bunch of stuff in sort of more simple slides just to make the point. But, we've had a lot of advances in how we examine, clinically examine, genetic etiologies of disease and conditions. We used to just use the basic karyotype to look at changes in the number of chromosomes. That's moved now to using microarray technology to identify duplications or deletions in the chromosomes that are up to 50 or 100 kilobases in size. And then of course now even more clinically common is actually whole exome or whole genome sequencing, where we can identify changes at the level of the single base pair, which helps us to identify single gene disorders. And with that technology, what's happened is that, and this is just a rubric, is that we've actually identified not just these big single gene disorders like tuberous sclerosis that we've known about for a long time that cause neurodevelopmental disabilities, but also copy number variant syndromes like 15q duplication syndrome that you'll hear me talk about a bit later, as well as these very small single gene mutations. And now taken together, these don't account for all of autism or all of neurodevelopmental disabilities, but the number is increasing in terms of the number of kids or even adults that we identify that have these putative causes. And what's important actually and interesting now is that with all the research done in sort of autism and neurodevelopmental genetics, it's now actually translated to clinical care. And now genetic testing is really the only routinely recommended medical workup for an individual with autism or, intellectual disability. And the clinical gold standard is that we do a microarray and then some other testing based on the sex of the child. Whole exome sequencing is often performed if the microarray is negative, because remember the microarray identifies copy number variants. It will miss single gene mutations and other kinds of very small variants. And so exome is performed. There's been a bunch of meta-analyses that I'm not going to show here that have looked at the clinical utility of whole exome sequencing and has found in certain cases where there's a high pretest probability that the yield can be as high as 50%. So we're really moving into this place, and the next set of recommendations probably will include whole exome sequencing. So why is that important? Well, first of all, we've identified now that about 25% of individuals with a neurodevelopmental disability have an identified genetic cause. So we're starting to think about, can we learn something from these syndromic neurodevelopmental disorders? Because we know cause at this point, right? And then again, if we think about precision health, we've opened a big door. So before I sort of get into that, I want to just make a couple of important clinical points that I think are really relevant when we think about these kids with identified genetic causes. So one is that clinically, these kids do tend to be more affected. And I want to be really clear about that. Sometimes, people talk about, certain genetic conditions as being models for autism or models for neurodevelopmental disease. And I always am very clear to say, these kids with some of these really large de novo mutations or variants, they're very clinically affected. So they don't represent the whole spectrum, right? They often represent the end of the spectrum that are more maybe clinically challenged in different ways. And what does that mean? So these kids often don't just have an autism diagnosis. And I don't mean to say just to belittle the importance of that diagnosis, rather that they don't simply have the social communication challenges or repetitive behaviors. They also have global developmental delay, especially motor delays and hypotonia, often have comorbid intellectual disability, and importantly, often have other comorbidities, not just medical ones, but neurological ones like epilepsy. So in certain conditions, we're seeing epilepsy in up to 50 or 75% of children. And epilepsy is just having more than one unexplained or unprovoked seizure. So, these are kids who are a little bit more medically and clinically complex, but you could argue these are also kids where we're really desperate for treatments, right, to really mitigate some of these issues that occur over a lifetime. The other really important point, though, and again, has been a really huge source of advancement actually over the last many years, is that as these individual rare conditions are identified, right? So again, individually, if you noticed on the slide a couple of slides ago that I showed you, individually rare, meaning each one accounts for maybe at the most 1% of the whole spectrum. Together, again, they account for about 25%. But each of these rare conditions, what happens is with social media and all of these other platforms, these parents converge, right? They communicate and they start talking about how they can work together to really accelerate not just a clinical understanding of these various conditions, but also really accelerate research. And so all these different parent groups are called PAGs, patient advocacy groups. And what they have done over the last, I'd say, decade is really improve our efforts in not just supporting families and advocating, but in facilitating clinical care through developing centers of excellence around the country for specific conditions, as well as facilitating research. And this is where, again, some of you as data scientists might be interested, but one of the big challenges for these families is like, how do we collect common data points across all these different, families that are all over the world, and how do we put them into one registry? It seems like a simple problem, but I cannot tell you how many groups struggle with this. And now there's efforts through this actually group that you can see here, Agenda, which is a collaboration of all of these different PAGs with which I'm involved. And it's sponsored by the Autism Science Foundation. The goal is to try to have these PAGs talk to each other to try to develop some common tools. But they're really working on developing registries, developing biorepositories, and then ideally developing platforms for trials. And again, I'm going to show you some examples of that. But these PAGs have been really critical in moving this research forward. But the big data issue, or not even big data, just the data harmonization issue, is one that they're all still often struggling with. Okay, so back to the precision health, right? So what can we do in neurodevelopmental disabilities when we think about these genetic conditions? Well, we can start thinking about syndrome-specific treatment. So as opposed to a standard approach where you take the whole sample of neurodevelopmental disorders, you choose a random cohort and you identify, a drug that might be effective. Instead, we take a tailored approach and we identify these subgroups that are, again, genetically identified at this point, and then think about treatments that would be effective based on the understanding of the mechanism of action of the genetic change. And that's exactly what's happened in our field. And again, in the interest of time, I'm not going to go through the different methods. I'm happy to answer questions, but there's medications that are more targeted to certain pathways. There's gene editing methodologies like CRISPR-Cas9 that's been very exciting. And then most relevant right now are actually these antisense oligonucleotides. And so what is an ASO? It's basically just a small sequence, right, of RNA that can bind to the transcribed RNA and change basically protein translation. And so that technology has really been leveraged in the last few years to be able to affect the downstream effects of some of these genetic variants. And I'll give you one example, which is not even really a neurodevelopmental disorder, but it completely blew the doors open in child neurology, I'll say, which is the ASO therapy for spinal muscular atrophy. So this is a devastating condition that affects, the most severe kind affects kids in early infancy, where they don't have production of a really important protein called SMN. And that protein is very important for just the functioning of our motor neurons. And so when children have SMA, they don't have basically strength. They don't have any motor function. And so you can imagine that affects their respirations as well as their movements. And many of these children actually die in the first year of life. And so an ASO therapy was developed to be able to essentially unsilence the silenced SMN protein that is, again, typically silenced in our bodies, because again, they have a deletion of one of the SMN protein genes. And with that ASO, this trial that was started again many years ago, halfway through the trial, they found that it was so effective that they stopped the trial and made this an FDA-approved drug called Spinraza. And now we've infants who are receiving this drug who are living a full life. I mean, completely curative, right? So it just shows that these conditions that we thought were never going to be able to be treated are now able to be treated. And I want to be clear, I'm not trying to compare a neurodevelopmental disability to spinal muscular atrophy. I'm just giving the point that, with an understanding of underlying mechanisms, we can actually develop treatments that could be deemed as transformative, or if you want to use the word curative, in this case, I think we would use that for SMA. And what's happened now in the neurodevelopmental disability space is that these ASO and drug trials have been now launched in various conditions. And I'll just point out a couple. So Tangelo is a Roche trial, and it's an ASO for Angelman syndrome. I won't get into all the details, but Angelman is caused by a deletion of UB3A, which is a gene that's maternally imprinted. So in our healthy brains, we have a paternal UB3A that's silenced and a maternal UB3A that's active. And in Angelman, the maternal UB3A is deleted. So what this ASO does is it unsilences the paternal UB3A. And we actually just had our first at UCLA Tangelo patient get dosed this week. So incredibly exciting. Again, the data are obviously not out yet, but the goal is that we can at least slow down progression or even possibly reverse some of the neurodevelopmental manifestations of these genetic conditions. So this is the era we're in right now, super exciting. And I really am careful as a clinician not to oversell these opportunities because we don't know yet, right? But if you think about where we've come from where we were 10 years ago when we were making genetic diagnoses that sometimes felt academic, right? They helped us inform parents about why their child might have developmental delay or not have language or have epilepsy or have autism. Now we're at a point where we can say, hey, based on a cause and based on our understanding with preclinical models and everything else, the underlying sort of downstream effects of that genetic variant or change. Based on that, we actually might have a therapeutic that could really fundamentally change the way our brain developmental trajectories are going to progress. And that's a very different conversation that I have with parents now in some cases, right, than I did many years ago. And so it's really exciting. So as I get you all excited, I also have to then also be, the tale of caution, which is that as these therapies are created, we have to be ready. And what does that mean? We have to be able to have the data necessary to develop and design and implement clinical trials that will truly tell us if the drug works. And unfortunately, in neurodevelopmental disabilities, we've been stymied by clinical trials being deemed as failures, and there's a lot of reasons for that, and we'll talk about it. But, we've had trials where people have been excited about, drugs, maybe more, for the broader autism spectrum rather than for specific genetic etiologies, but a lot of excitement around trials that then in the end, the drug doesn't seem to be more effective than a comparison group. And then it's, never approved for use. And that really leads to some discouragement. But I would argue that it's probably not always the case that it's because the drug doesn't work. It might also be because we don't have the right trial design. So let's talk about that for a minute, right? So just to, again, this is really basic 101, and many of you probably already know this, but it helps when we think about, again, how we can, improve clinical trial readiness, which is, again, going to be the bulk of the rest of this talk. So how do our trials design? So typically in a trial, you take your initial population of interest, and again, there can be quite a bit of stratification that's done up front, but you take your population of interest and randomly split them into two groups, right? And then one group receives an intervention and the other groups receives a placebo. And I won't get into it. There's lots of different trial designs that allow for us not to have to do a pure placebo, like, sugar pill or whatever, so that people do always have a chance to get treatment. There's staggered wait list designs and things like that. But fundamentally, the idea with a trial is that you're asking whether the intervention group has made gains in whatever your outcome measures of interest are. Have they made gains above and beyond what we would see in patients who did not receive the treatment? That's the bottom line. So why are we challenged in neurodevelopmental disability clinical trials, especially in these syndromes? So the first is that we don't have great measures of drug target engagement, meaning we don't know if the drug actually hit the target before we decide to then launch into a one-year trial to follow outcome, right? So it'd be really helpful to have some measures of that. The second, and this is a big one, is that our standardized clinical measures that we use to assess development, cognition, social skills, they often don't adequately capture function or even change, especially in these populations that are more severely affected. And I'll show you an example of that. And then lastly, as you can imagine, these syndromes are rare individually, right? These kids are all over the country and the world. So how do we actually get a sample size big enough, right, to be able to answer the questions that we need to answer when you can't just recruit locally? right? These kids are everywhere. So those are, I would say, the three really big areas of challenge. And so we in our group have actually been doing research to try to address all of these areas. And what I'm going to do in the next like 15 minutes or so is kind of show you examples of what we've done in these three areas. And again, each of them could be their own talk in the interest of time. And also just to give you kind of high level view, I'm just going to show you examples, but happy to answer more questions about them. And a lot of the stuff is published or recently published as well. So, okay. So the first is this issue of drug target engagement, right? How do we address that? And so the way that we've sort of tackled this, and again, this is not just my group, this is the groups around the country and world, is that we can start thinking about how do we identify mechanistic biomarkers, right, that really inform drug target engagement. So biomarkers that we think reflect the putative target of the drug, and that we also think reflect maybe some of the mechanism underlying disease or disability in these patients. With regards to this issue around the challenge around standardized clinical measures, we've been doing quite a bit of work thinking about and developing through big cohort studies, through an evidence-based approach, what are the most meaningful and measurable clinical endpoints? So we need to test those clinical measures in these populations directly to then be able to answer the question about test-retest reliability or stratification and those sorts of things. And then lastly, from the standpoint of the challenge around the rarity of these conditions, we've been working to design protocols and develop methods that really maximize access and scalability. And this has become amplified during COVID, because forget about whether someone has a rare condition or not, these approaches that we take to figure out improved access are going to be relevant across the board, because right now families can't travel even if they are local because of restrictions. And so we've had to be really creative with that. And I'll show you a bit about that at the end. Okay, so let's start with this issue about biomarkers. So what is a biomarker, right? So a biomarker is just any characteristic that's objectively measured and evaluated that indicates either a normal biological process, a pathogenic process, or a response to treatment. There's a lot of interest and excitement about biomarkers in autism right now. And I'll just mention that there's a large NIH-funded Autism Biomarkers Consortium for Clinical Trials, of which we are a part. Jamie McPartland at Yale is the PI. And our goal is to use methods like EEG and eye tracking to quantify some biomarkers that will help us stratify kids with autism and might be sensitive to change over time. So this consortium study is not a trial itself. It's really a big data gathering initiative. And some of those papers are getting published right now from our first wave. We just got refunded to start again this summer. But you might ask, well, what kind of methods and what kind of biomarkers do we use for a neurodevelopmental disorder? Well, this is again where genetics can help us think about it. And this is kind of a busy figure. So if you have a small screen, you might not see it well. This is a great review by Luis de la Torre from Nature Medicine a few years ago, where he basically looked at kind of what are the convergent mechanisms and pathways that underlie these like hundreds of putative genes that cause autism and neurodevelopmental disabilities. And it turns out, not surprisingly, that the many, many genes that are affected and that are causative really do impact these basic neurodevelopmental processes like protein translation and synaptic signaling. And that these effects and the initial effects of these genetic changes occur before birth. So this circle is birth, right? So these are coming online in utero fetal brain development. And fundamentally, again, as I mentioned, they're affecting things like synaptic signaling, gliogenesis, migration, differentiation. And when you look even sort of at a slightly sort of macro scale as to what these different pathways actually do affect at the level of brain function, they fundamentally affect brain circuit function, right? So they're affecting the way, as I like to think about it, the way the brain is wired, right? So it's not that these, that we're seeing a break or a break in healthy connections that have already been formed. It's that these sort of network level, changes that are supposed to occur in typical development are just not forming in the same way. And that actually led to this coining of the term by Dan Geschwind and Pat Levitt many years ago that autism and neurodevelopmental disorders were developmental disconnections. So again, it's that these connections are just not forming in the way they might form in typical development. So how do we test that, right? And so again, a bit of an oversimplified slide, and if any of you are electrophysiologists, you might appreciate this, but we've been using EEG as a really beautiful, temporally sensitive tool to really assay neural circuit function in real time, right? So we can really probe that circuit with scalp level detection. Again, this is a picture of the kind of nets that we use, but there's lots of different ways that we can collect these data. And fundamentally, what is EEG? It's really measuring at the scalp the synchronous firing of neurons in real time, right? So it's a fairly nice, somewhat indirect, although I would argue not so indirect measure of healthy circuit function and development. And there's so much that we can do with the signal. And my lab has published a lot of EEG papers. I'm not going to go into all the methods, but just to say that kind of crude signal we get at the scalp, we can break down into component parts and calculate, the oscillatory power in different frequency bands. We can look at peak frequency, and we've done a lot of work looking at peak alpha frequency in particular as a very sensitive measure of really healthy brain development. And we also can infer connectivity. So unlike with imaging, where we can really look nicely at structural connectivity, with EEG, we cannot do that. But what we can do is using tools like, and signal processing methodologies like coherence, we can ask how in sync is the signal in different parts of the brain. And by doing so, we can infer how connected different parts of brain regions are. Okay, so that, and we have focused quite a bit on alpha phase coherence in our lab. And so using EEG as a method, we can really start to ask these questions about, are there robust biomarkers in these neurodevelopmental disorders? And I'll give you one example of the infancy work again, and this is again a talk unto itself. I didn't want to focus on that today because I'm trying to tell the story around clinical trials. But we've actually for a long time been studying infants who are considered at higher likelihood or higher risk for having a neurodevelopmental disability, either because they have a genetic syndrome or because they have an older sibling with autism. The baby siblings group is one that's been studied for a decade. And Jack mentioned at the beginning that we have a large international consortium actually of researchers who've been studying baby siblings. And I've been chairing that consortium just for the last year. But, there's been quite a bit of work in that consortium using methods like EEG to study early brain development. And so this was a study that just got published, and Abby Dickinson, who's a really phenomenal research scientist in the lab, worked with actually a bunch of data scientists on this and took kind of I like to think of it as like a clear box machine learning approach, where they basically asked, which networks, which set of networks, which we called features, and the networks were defined again by phase coherence between two electrodes, essentially, which networks most robustly relate to and predict later ADOS scores, and ADOS is a clinical measure of autism. But the nice thing about the way that they did this algorithm is they were able to then go back and look to see what the networks were so they could be interpreted. And what we basically found was this pattern of hypoconnectivity or reduced phase coherence in frontal cortical regions, and a pattern of increased connectivity, increased phase coherence in temporoparietal regions, which is interesting because those are networks that have been for a long time in the imaging work been implicated in autism, right? Frontal cortical regions being really critical for attentional networks, especially social attention, whereas these temporoparietal networks have been more clearly linked to really social cognition and social motivation. So again, really interesting work, just got published, and I'm happy to share that paper with you guys. But our next step is really to replicate this, right, in a larger sample. But this shows that, and sorry, I didn't mention this is in 3-month-olds. So, this shows that we can use EEG to kind of map these changes very early in development. How robust they are, how predictive they are, definitely still needs work. So I don't oversell the finding, but I just want to show that we can use EEG in these ways. Okay, so back to sort of thinking about these syndromes and how EEG might be a useful biomarker. We've been studying a bunch of these different, really highly penetrant, for neurodevelopmental disabilities, genetic syndromes. And one in particular we've been really interested in, and I think it just is a good example of the kinds of ways we can use biomarkers to study these conditions, is Dup15q syndrome. So Dup15q is a syndrome caused by a duplication on chromosome 15q. You can see here the region that's duplicated, and there's a bunch of really important genes for brain development in this region, including the maternally imprinted UB3A, which I mentioned earlier, that's deleted in Angelman syndrome. In Dup15q, it's overexpressed. And there's also a cluster of GABA receptor genes. And that's important because we do really believe that GABA neurotransmission is profoundly altered in these individuals. And what we know about Dup15q syndrome is that it's highly penetrant for neurodevelopmental disabilities. So these kids have hypotonia, global delay, autism, as well as epilepsy. What got us actually quite interested in this condition is that there was a case series that was published many years ago where they were describing the clinical EEGs of these children. And, these kids get clinical EEGs, undergo clinical EEGs because they have epilepsy. So they need to get EEGs for monitoring. And what was found was that these kids had this sort of high frequency oscillations on their clinical EEG. Now, as a neurologist, when I see this EEG pattern, it makes me think about that the child's taking a GABA positive allosteric modulator like Ativan or Valium. So when you actually increase GABA signaling with pharmacologically, you actually get these high frequency beta oscillations. These kids are not taking these medications. So when we saw this, we thought, oh my goodness, this might be a biomarker of that GABA, those GABA genes being overexpressed and altered GABA neurotransmission. So let's try to really quantify and study this biomarker. And that's just what we did. And again, this has been published over the last few years. This was great work done by a couple of amazing graduate students and a statistician named Damla Senturk. But what we found using our signal processing methods is that in DUP15Q, this is just sort of like a heat map, a topo plot, just in the beta band range, which is like 15 to 30 hertz, we see much higher beta activity in DUP15Q compared to IQ matched kids with non-syndromic autism and typically developing kids. This is a power spectral density plot also just showing this big GABA peak. And this was huge. The effect size is almost 2 here. Scatter plot showing the same thing with the high beta in the kids with DUP15Q. Since then, we've done quite a bit of work that was recently published asking about whether it's stable over time, that biomarker. The answer is yes. Whether it's scalable, can we measure and quantify this, not just in our research EEG, but also in our clinical EEG? Answer is yes. And it's also led to this very interesting path that I'm not going to get into today, but if you're interested, once it's published, I'll send you the paper. We just submitted it, where we started looking at EEG and sleep, because I was wondering if sleep physiology was altered, given that we see these high beta oscillations in the awake stage. And I thought if sleep physiology is altered, that might have a profound implication on cognitive development. And so we went down this pathway and found actually that there is profoundly altered sleep physiology in these kids, not just in the fact that they have beta at night, but in their slow wave oscillations and their spindles. And we think that's going to be a really important actually target for therapeutics. So that's an example of a really nice biomarker, right? And so taking it back to this rubric of precision health, let's just sort of operationalize this before we talk about the clinical endpoints, which is kind of the next area, which is, imagine that we see a child with DUP15Q syndrome, right? Or has symptoms, right? Global delay, autism, some seizures, and we get the molecular diagnosis of DUP15Q. Well, imagine now that we actually conduct our EEG assessment, we find this beta biomarker, and then we have the opportunity, and this is literally in the works right now, that there are drugs that are GABA negative allosteric modulators that actually can reduce that the overexpression and the excessive GABA signaling. Well, then those beta oscillations might be a really robust marker of drug target engagement, to know that GABAergic treatment, if you will, that modulator did hit the target. That's exactly what we're in the midst of designing right now with some trials. So very exciting, and it's really accelerated again and changed the way we're thinking about how these biomarkers can be used in trials. Okay, so the next part that I mentioned was just that, so now we have these biomarkers. Now, what about meaningful endpoints? How do we even think about that? I'm not going to spend a ton of time on this, especially because I know a bunch of you aren't like clinicians, so I won't go into the weeds on this, but I do want to show you this, because I do, again, think as data scientists, this is relevant, because we really struggle with sample sizes and thinking about how to harmonize data in these ways. So we, with the same syndrome, DUP15Q syndrome, we wanted to really understand what is the clinical phenotype and how do our measures of cognition, motor skills, and adaptive skills, how do they behave in this population? Because we're getting ready to design a trial, we want to know what our endpoints should be. And so going back to the slide I just showed you, what we did was we didn't just collect data at UCLA, we actually took our team on the road and we went to multiple family meetings where we could collect phenotyping data. We actually did EEG assessments there as well for some of the papers that I mentioned earlier, because it was the most effective way to get access to these families. And they were very responsive to that. So what did we find? Okay, so one of the first things we found when we actually did our battery of assessments, and we also compared these kids to non-syndromic autism, is we found that these kids have really significant motor impairment compared to non-syndromic autism. This is on the Vineland, and this is on the Mullen, if you're familiar with those measures. So this is a parent report, this is direct assessment. So big picture, significant motor delays and motor impairments above and beyond what we see in non-syndromic autism. This actually has been corroborated in large cohort studies, like in the Simon Simplex collection, which some of you might know about, where they also did a study where they looked at various measures in kids who had causative genetic, syndromic, basically autism compared to non-syndromic. And the biggest differentiator was age of walking. So later age of walking in kids who had causative genetic variants. So we asked, well, our motor measures are really crude. Can we develop more quantitative ways to assess motor skills? And I don't take credit for this work. This is a fantastic junior faculty member in my lab who was a postdoc with me, and now she's a K23 to really explore this. She used gait mat technology to better quantify deficits in gait in kids with DUP15Q. This is actually a kiddo with DUP15Q at one of the family meetings. They took this huge gait mat and schlepped it to a family meeting to collect data. And you can see that kids with DUP15Q have different gaits than kids with non-syndromic autism. And specifically what this Dr. Wilson found was that these kids have slower velocity of gait as well as shorter step length. And this is a nice measure, right? This is something that's quantitative. You could argue it's almost more like in the biomarker category than clinical assessment, but think that this is something that we can use to measure if we think that some of these drugs will actually improve motor skills, probably in a way a little less crude than some of our standardized measures. We also, though, did, and I won't spend a lot of time on this, we did collect, again, a lot of standardized data. And you'll see where the challenges are. And again, for you guys as data scientists, I want to show you this so you can think about how do we think about solving some of these problems. So this is just a cognitive assessment called the Mullen. And we just did age, we call them ratio IQs. So it's really the age equivalence over the chronological age. And even using that kind of ratio IQ score, you can see, and again, 100 is considered, quote, normal. Right? Look how low these kids are. Many of these kids are at the floor, particularly kids with seizures. So kids with seizures are more affected than kids without. That's sort of a known phenomenon in clinical medicine. But we have kids who are really falling on the floor. So how do we use those measures when we want to maybe target cognition as an endpoint for a trial, right? Interestingly, though, when we actually look at other measures of functioning, like the Vineland, which is a parent report measure of adaptive skills, we see a bit better stratification, and kids aren't quite at the floor as much. Right now, that might be partly a bias of parent reporting, or it might be that measure is actually just collecting data that is a little more sensitive to subtle abilities that these kids have. But we like these kinds of measures that have a histogram like this versus one that has a big left shift. But we have to contend with this because these are the measures that we have to work with, right? And so, we've based on that work, and this paper just got published, actually, we've been really thinking about how can we be more flexible in studying and testing cognitive abilities in these kids. There's other questionnaires we can use to look at comorbidities and social skills like the SRS and the CBCL. And I'll tell you that we struggled with those measures for these kids with more significant disabilities because some of the questions that are asked of parents are just not answerable. So for instance, on the SRS, and it's a great measure, the SRS has a lot of strengths and value to it. But, some of the questions are things like, your child's aware of what others are thinking or gives unusual reasons for doing things. Well, if a child's nonverbal, that's going to be a difficult question for a parent to answer. So then we get a lot of missing data. So we have questionnaires where we're missing a third of the answers because parents don't know how to answer it. How do we deal with that? How do we use that as a measure of outcome? Right. So those are sort of issues we're still grappling with and ones that hopefully some of you can help to address. But taking us back to now this rubric of precision health again, we talked about the biomarker in DUP15Q. Well, now based on some of the studies we've been doing with our larger cohorts, we can start being a little bit more precise with thinking about outcome. So you might not use a ratio IQ score or a full IQ. We might use quantitative motor skills. We're definitely going to be excited about asking about adaptive skills. And maybe we'll use raw scores for our developmental or cognitive assessments because even one behavior or ability that's gained might be meaningful, right? So, but we're going to really change the way we're thinking about outcomes based on the kind of data that we're collecting. And this work is ongoing, and there's a lot of sites around the country that are sort of collaborating on thinking about these challenges. Okay, good. I'm doing well with time. So the last couple of pieces I'm going to try to be done in about 7 or 8 minutes, and then I'll have time for questions. So lastly is this issue about the fact that these syndromes are rare. I'm not going to spend a lot of time on this one. I just want to emphasize that we are working hard as a field to develop protocols that are more accessible. And I'll give you one example. So we've been studying another rare condition called tuberous sclerosis. Details don't matter actually for the purposes of what I want to talk about. What I will emphasize is that TSC is often diagnosed in utero. These kids have these benign hamartomas or tumors in the brain. This is a fetal MRI, as well as in the heart. That's the fetal ultrasound that are picked up early. And what we also know is that more than half of these children end up being diagnosed with autism. So we've been over the last 10 years studying these infants prospectively from early infancy to ask, are there early predictors that we can then target? Right? Can we start getting these kids into early intervention early on? And what we found, and all this work has been published, this is in the blue box, we have found that by 12 months of age, we can identify pretty robustly delays in nonverbal cognition, social communication skills, and actually with EEG, we've identified biomarkers of atypical development, both in peak alpha and in phase coherence that relate to later neurodevelopmental outcomes. So we have these robust measures, right? And so what's the obvious next step? Well, we need a treatment. And so we designed and were funded a couple of years ago through NIH to run the first randomized controlled trial of behavioral intervention for infants with tuberous sclerosis. And this was an intervention called Jasper that was developed by Connie Castry at UCLA. It's a really beautiful intervention that really targets some of the core symptoms that we were identifying in TSC. So it targets joint attention and social communication skills. And so our question was basically, do infants who receive weekly Jasper, do they show better neurodevelopmental outcomes than those that do not? And we had like a waitlist control design. So everyone eventually gets Jasper, but some kids have to wait 3 months because we felt like ethically we wanted to make sure everyone had a chance to get the treatment because they'd never had access to this before. And the way the study was originally designed was that they were coming in every week to either UCLA or Boston Children's Hospital for the intervention. There was a lot of excitement around the study when it was launched. And, social media and the press and the patient advocacy group, everyone was getting excited about this. And in our first year of funding, this is an R01, so this is not a trivial amount of funding, this was our enrollment table. So yellow were the number of families enrolled. So you can do the math and realize that is not enough families. Our goal was 60. But the purple dots were people who were interested but didn't enroll. So you can imagine why, right? Or maybe you can't, but what might be an issue, right? Look at that map, right? These kids are all over the place. And essentially parents said, we just can't do it. It's not convenient, basically. And one parent actually said, if virtual ever becomes an option, we'd be interested. So we went back to the drawing board and we basically converted the entire Jasper study, which is called JETS, Jasper Early Intervention for TS, to a remote delivery. Parents still have to come in once at baseline for the EEG and the assessment. But after that, what we do is we give the parents an iPad and they get trained once in person. This is just a picture. In the interest of time, I won't show the videos, but the examiner or the interventionist teaches the parent in real time about the intervention. And then the parent goes home and practices. And every week they upload their video on their iPad to Box. It's very archaic, but it works. The interventionist then downloads it from Box, reviews it, and then they do a Zoom call once a week. And this was pre-COVID that we started this. Every week there's a new module. The parent gets trained again remotely. They practice, same cycle. And so the whole thing is parent mediated, done remotely. So we made that change. And within 6 months, this was our enrollment map. And we've continued to enroll. We're not done with the trial yet, but it's been super exciting. And I think and we just published actually that paper to describe the methods. And there's lots of studies now. This is an Angelman natural history study that's all remote, right? Or mostly remote with in-home assessments, because we all recognize that it's hard for families to come, especially when they're geographically dispersed and have kids with complex medical issues. So that's a great example. And we're doing a lot more work in that area now. Okay, so I'm going to end with like 2 minutes on this because I just think it's an important thing for us to all sort of be on the same page about, which is how have we had to modify things and how we had to think about these families in the era of COVID. And I'll highlight a really nice letter to the editor that John Constantino wrote with Joe Piven and some others recently about, just the impact of COVID restrictions on individuals with IDDs. And what they said, this quote I thought was really powerful, and I see it every day when I see patients in clinic, which is that among non-infected persons in the US, there's few more adversely affected than individuals with IDDs. And why is that, guys? Well, these individuals, they rely on intensive services, especially in home services, right? That they're not able to get now. They need more close medical monitoring, which is also hard to implement remotely. They're also more overall vulnerable to the impact of the educational mitigation efforts, right? Not being in school during the day. Zoom is very hard for a child who has significant intellectual disability or has autism or limited attention. And so this is really important. And so we actually did a survey to really understand the effects of COVID on individuals with IDDs, NDDs. And the first survey was done in April, and that was already published. And we're doing a second wave actually now. But just some highlights. And again, that paper just came out, so I can share it with you, or you can find it on PubMed. But, important facts, like more than half were unable to see at least one healthcare provider. 3/4 lost access to at least one educational services. And we had a handful, about 30% had lost access to all their services. But, interestingly, when we asked parents, what would you like to see, preserved, access to clinical trials is one of their biggest priorities. Like, they want to still be in trials. They want to still be involved with research. And so we have to think about how to keep moving forward, even though we have these challenges and these restrictions. And so we've made a big shift in a lot of our research in clinical care. A lot of the stuff that I described that we're doing, the biomarkers work, the clinical endpoint work, we're still doing it. We're just having to be more creative, right? So we're incorporating telehealth quite a bit more. We're working on validating remote behavioral assessments. And this is a big consortium-wide effort with the BSRC and some others. We're figuring out ways to collect biomarker data remotely through signal processing of clinical EEGs, even portable systems. And we're just thinking about how do we do all this while preventing this widening of disparities that inherently occur for parents who don't have access to technology the same way that some of our more resource parents do. We have been bringing kids back to the lab for EEG though, and I'll show you some of the things we've done. We moved our entire control system outside into the hallway. This is my one hallway of my lab. All these doors are EEG testing rooms actually. So this is one of our rooms. The parents on the inside with the kid, they become the behavioral assistant. So we have no one in the room with the child. So the parent does everything. We coach them ahead of time, send them a video and all kinds of documents to help them get ready to prevent their child from pulling at the net and those sorts of things. Oops, sorry, I hope you can still see my screen. But our control, system is outside. We have a plexiglass wall here so that if there is an examiner on this side, they're at least separated a bit from the family because the kids are not wearing masks, usually just the parent is. But we're doing a lot of these different changes, and I can talk to you more about that now that I'm done. So I'll just end with this, right? So we talked about precision health, right? Delivering the right treatment to the right patient every time at the right time. And, I just say that right now in COVID, we also have to think about how do we do all that safely and accessibly. And that's been sort of something we've spent quite a bit of time contemplating and persevering through over this last few months. And with that, I'll stop and just say thank you for listening. We have a bunch of funders that we appreciate greatly. This is my amazing lab pre-COVID. Now we're doing a lot of things on Zoom. This is actually a group of students who are working with me this summer. And we had to coach and mentor students completely remotely this year. So that was, I'm sure you're going through the same thing. And it's definitely a learning curve. So with that, I will stop and I'm happy to take questions. Okay, should I stop share? Are you guys?
Hi, Shefali. Sorry about that. I just needed to unmute myself and couldn't find the button. Thank you so much this is a fantastic and wonderful talk. This is just very, very well done. It is a tour de force amount of work. And so congratulations to you and your team for all of it. I want to start off by asking you a quick question, which you kind of covered. You covered biomarkers and EEG and genetic testing and cognitive assessments, et cetera. You talked about some of the treatments that can go on and some of the effects of those treatments and their effects on cognitive and motor results and in clinical endpoints and hopefully cures for some of these sort of disorders. One of the questions I wanted to start with is, can you discuss some of the ethical issues involved in the start points? We want to get to endpoints, obviously, but where do you start? And you kind of mentioned children being genetically tested in utero. You mentioned, some other things about, when these things would start in childhood to hopefully kind of head these things off of the past. What are some of the ethical issues about this that you've encountered and that, people discuss in the field that people might be interested in knowing about?
Great question, Jack. And I think there's actually a few junior investigators in the BSRC, mostly working with IBIS, which I didn't mention today, but IBIS is the Infant Brain Imaging Study, which is a imaging consortium of infant siblings. And I have actually an R01 that actually has added EEG to IBIS. And so we work quite closely with them. There's a whole subgroup that's been developed on this question about the ethics of early screening and detection. And so there's a couple of things. So first, if we think about just the early screening detection, forget the genetic testing piece first. It's just, what are the ethics around, identifying early risk markers, right? And when I say risk markers, I mean, and there's a lot of controversy in using the word risk. So I'm using it as a clinician, meaning I'm thinking about, identifying markers that herald high likelihood of having a neurodevelopmental disability later, right? Which comes with, challenges and a lot of cost and other things for parents and healthcare providers over the years. The question becomes, if our predictors aren't 100% accurate, right? That's one. And two, if we do not have actual treatments for all of these conditions, if you will, especially if it's polygenic risk or not a known genetic cause, then what are the ethics behind that, right? Is it ethical to tell a parent, your child has a 50% chance of having autism when there's nothing that we can actually recommend from a treatment standpoint to change that trajectory? And the answer is we don't have a great answer to that. There's a lot of dialogue about this. There's a lot being actually written in the field about this. But so the argument pro screening is that simply by more closely monitoring and providing just the basic behavioral interventions that are available, we do believe that we improve outcomes compared to what we would do if we never had that early screening and surveillance going on. So those are the pros, right? The cons, though, are that, it's adding stress to families, right? We're telling parents, well, there's a good chance that you're going to have more challenges, but we can't tell you for sure. And we know that parent stress, family stress, those also are not always protective, right, of healthy outcomes. So there are absolutely arguments pro and against. And we get asked this question when we write grants in this area also, like, how do you mitigate the stress that you're causing families by, with this kind of work where you're just following and screening so closely? So it definitely is a challenge. I would say that, then the added question around genetic testing, now there's prenatal genetic testing available, through maternal blood, we can do it, right? And you can do microarrays and other things. And we are not at a point where we have gene therapies or therapies for all of these causes, nor do we know 100% what the outcome will be in some of these very rare variants. And so there's a lot of controversy over the clinical value of performing this kind of genetic testing very early. So I think we're at the epicenter of these debates, and I think it's going to be a really interesting, next 5 years as we sort of really sort out how we handle that.
I think you're absolutely right. Some of these questions are, the technologies are possible, right? Yeah. But some of the ethical questions about when to apply them, at what point in development, or, who knows, if you wait, you don't see something in a child, you wait 2 weeks, maybe it'll show up. But if you do something before that, you could, actually cause more harm than good. One thing I wanted to ask you about from some of our participants is, do you think any of the approaches might be effective perhaps as protective treatments for neurodegenerative disorders affecting older adults? Any comment on that?
Great question. And I think that the answer is yes, I think it is possible. There's sort of this, again, and my mentor, Chuck Nelson, has been a part of these debates about, are there critical periods in development? Are there just sensitive periods? Are there times that, is there a point in development where it's too late? Right. And I don't think any of us believe that at all. I think that thus far, there just have not been robust enough trials in adults, especially with some of these more what we would call disease modifying therapies, to know if we would see a change or not. So I think it's worth exploring. I do think, though, like, word of caution is that, kids are not just little adults. And so the kinds of measures we're using, the expectation of change, even our biomarkers need to be different in our young populations compared to what we might expect to see in adults. But I think that the adult space is, and I've actually the Abby Dickinson, who did that who wrote that paper that I showed you on the kind of machine learning with EEG in our high-risk infants. She's actually writing a K right now to look at sort of aging in autism and to ask exactly this question. So the measures that seem most sensitive to cognitive development, for instance, in early infancy, are those also sensitive to aging processes? Might they predict neurodegeneration or aging in autism? So, so that's an exciting area. Kathy Lord's going to be her primary mentor, and they're working on that right now. So, so more to come there, but it's a good question.
That's really cool. And well, another question is, as people are looking at these various biomarkers and looking at these profiles across different disorders, whether it's autism or other, developmental delays or, any other sort of things, are people seeing some sort of commonalities across them that would suggest that, there's actually fewer independent diagnoses than there, we might have carved the population up into? Does that make sense?
Yes, and actually I saw that question in the chat, and what I will say is that, and this is for all of you data scientists, I want a figure like this with EEG. So my point, I would love to see, and it's not going to be it's going to be complicated because EEG, it's fraught with variability. I'll just say that, right? But, yes, I think that convergence at the level of genetics, but there's going to be convergence at the level of sort of macro level circuit function. And yes, I mean, there may be other kids the simplest examples, there may be other kids who have high beta that might be amenable to similar treatments that might be effective for Dup15q, for instance, right? But we need, the issue as, again, and I sound like I actually didn't say it a lot in this talk, I say it all the time to my lab and to others, is we just need big data, right? Like we can't identify patterns and subgroups with sample sizes of 50 kids. With a range of we need. And so, I've been working, one of my big goals at the BSRC was to try to harmonize EEG data across all of our sites that are collecting EEG. And we haven't really gotten there yet. And there's just, it's details as to why it hasn't happened. The IBIS study that I have that I didn't mention, the IBIS grant that I have, this is Joe Piven's study where they're prospectively imaging babies, and it's a substantial sample size of 250 infants. So we got a grant to add EEG to that study. So at the end of the 4-year IBIS study, we will have EEG prospectively collected and imaging on 250 infants. So that will be, I think that'll give us some nice, that'll hopefully get us in the right direction here. But really, we would ideally want sample sizes that are even larger, to really ask, are there EEG patterns that help us select kids for trials?
Well, with that, Shefali, thank you so much for sharing the work of your laboratory, your thoughts and vision for what you would like to see in this space. And I completely agree. Large-scale data across multiple biomarkers and across multiple constituents of our population are really going to help get some insights into some of these developmental disorders as you described. So thank you so much. I appreciate it. Everyone, thank you. Enjoy your weekend, and we'll see you again next week. Thank you so much.
Awesome. Take care.